Okay, so good afternoon, everyone. I hope everyone has enjoyed their tea or coffee or milkshakes or juice. And welcome to the last session for the day, which is on planted pastures and feeding ecology. So we have 12 speakers for this session. 2 of them will be 15 minutes, and then the rest, the 10 of them, will be short presentations that are 5 minutes. So may I plead to the presenters to please keep to their time that has been allocated to them so that we can continue the program in time and be also in time for the AGM that is later this afternoon when we are done with this session. The timer, if you are standing here on the podium, the timer is there at the bottom on your right-hand side. You will see how much time you have left. And for today, the person who is starting our session is Dr. Thamsanqa Mpanza, who will be talking to us about in vitro ruminal fermentation and methane emissions of Eragrostis curvula and grass supplemented with Searsia. So the floor is yours, Dr. Mpanza.
Thank you. Thanks, Program Director. I'm not going to spend time reading a topic already, it has been shared. Now, since from the morning we've been talking about different species, let us deviate a bit just to look at the digestive system of an animal, especially a ruminant. The key critical component in a ruminant animal is this one, which is called a rumen. Why? It's because the microbes that are there produce 70 to 80% of the energy that is required by the animal in a form of volatile fatty acids. In addition to that, it produces 70% of the daily rumen or daily required enzymes which assist in fermentation during a fermentation process. In addition to that, it also produces almost 90% of required protein for production. Now, because of the microbes that are there. However, the problem is the production of methane, which contribute to global warming. Hence, ruminant animals are blamed for contributing in climate change because of methane. In addition to that, that methane is a reduction of is energy loss, which negatively affect the production. Therefore, the question is, how can we modulate the rumen in order to reduce methane without affecting fermentation? Hence, the objectives of the study was to evaluate the effect of supplementing Eragrostis curvula grassae with Searsia lancea, with an aim, with a focus of in vitro study, with a focus of gas production, methane emission, as well as the rumen fermentation. We use Eragrostis curvula as a substrate with a pH or with a crude protein of 5.334, which is far less than what a minimum requirement of 7 for just maintenance. And the Searsia lancea leaf meal that was used was shade dried, and the other one was ensiled. But what is interesting that we want to look at, when we ensile Searsia lancea, the protein content increased to 14.22 from 10.68 on a shade-dried material. We thought perhaps since now it is increased, how effectively can we use that protein in order to boost the poor quality protein of the Eragrostis curvula? Then we supplemented it at 2 levels, 15% and 30%. Then we look at in vitro gas production, which was collected in 3, 6, 12, 24, and 48 hours. In 12, 24 to 48 hours, we collected some samples of gas and we analyzed the methane. At the end of the 48-hour incubation, we look at the volatile fatty acid as a means of indicating the fermentation. Now, in this slide, it shows that the grass that was supplemented with leaf meal from the dried composition, you can see that it's improved gas production. which means that it enhances the digestibility of the grass. However, what was interesting is that when we applied the one that was ensiled that had many protein, we only see high protein rather, we only see significant increase at 3 hours when we use 15%. Other than that, there is no increase, which triggered a certain question that we needed to follow. This is what I was discussing basically. But then we concluded that supplementing grass with, we decided that supplementing grass with silage leaf meal at 15% is effective only for 6 hours. Then when you look at the methane production, which was our main target, you can see that at 12 hours, the methane gas is more or less the same. There's no significant difference, though numerically, there's more methane produced here. But when you look at 24 hours, it becomes significantly increase on a grass hair alone. The reason is, remember that the protein content was far less than 7%, which is the minimum required protein level that triggers the normal functioning of the microbe in order to ferment the material. Hence, the protein, the feed will take longer in the rumen in an animal, which lead to exposure of that microbes trying to break the material so that it can digest it. In that process, then it's lead to methane emission as the time goes by, as you can see there. Now, the high protein produced in 24 hours and 48 hours could be attributed to the delay fermentation due to the low protein content, which is far below than the level that triggers the microbes activity. Here we look at the in vitro fermentation as a means of indicating to say, adding this or supplementing Eragrostis grass, does it affect the fermentation, which is the volatile fatty acid production and its organic gas? You can see that none of them was affected. It shows that none of the supplement affected the rumen fermentation. So why is it so? Because if volatile fatty acids are the product or immediate of product in the rumen, which determines the energy production during fermentation. At the same time, it can lead to the hydrogen production, which that hydrogen is used by archaea bacteria to form methane. So now a question perhaps that might arise would be, if you look at the grass that is supplemented by leaf meal that was dried, it reduces methane. So we need then to understand the logic behind to say, this supplementation, does it affect the gas production? Obviously, no, not. But what does it affect? So we had to look at for example, employ the metagenomic analysis where we can collect the DNA sample and do the bacterial composition. We can able to see what bacteria that dominate in each of these treatments so that we are able to perhaps predict what happened. The other thing that is interesting is that when we ferment when we ensile the Searsia leaf, it's resulted to the increase of methane significantly so compared to the one that was shade dried. We thought because of that more protein extra that is produced, which we assumed that is because of perhaps the reduction of tannins, which is sensitive to pH, but it seemingly shows that, so it seems to be indicating to us that possibly a certain compound might have taken over, which prohibit the availability of that protein for the animals, which then therefore we thought a metabolomic study will be required in order to characterize both material or both leaf meal so that we are able to see if the tannin reduce and release more protein, what else comes over and what is the bioavailability of that. Thank you.
Thank you so much, Thamsanqa Mpanza. Before you go, I have time for one question in the audience. So if there's any burning question for Dr. Mpanza, you can raise your hand. Going once, going twice. released. Thank you very much, Dr. Mpanza. Can you please give him a round of applause? And then our second speaker for today, for this session, is Sigrun Ammann, who will be presenting to us on evaluating red clover and the link to system fit in multi-species in dairy pastures. All yours.
Good afternoon, everyone. Okay. Okay, so we use multi-species pastures now quite extensively for dairy production. And the reason for doing that is we try and achieve improved resilience in these pastures. We try and improve the productivity year-round. And we try and achieve greater environmental sustainability through planting these multi-species pastures. So if we look at that in a little bit more detail, the resilience is mainly to try and adapt to climate change in our area in the Southern Cape, that is mainly temperature and water availability. The main reason to try and improve productivity is to improve the fodder flow throughout the year and to improve the forage quality throughout the year. And for environmental sustainability, we try and reduce the nitrogen inputs, for instance, generally inputs, but the main would be nitrogen, and improved water use efficiency, which mainly links to the deeper rooting systems of the species we use. In multi-species pastures, it's very important to look at complementarity of the different components. So for instance, that would mean looking at different types within species that are represented by different cultivars. So today I just want to focus on red clover because that is quite an important component of our multi-species systems for intensive dairy production. So until recently, the only red clover cultivars available in South Africa were actually the annual types. And that is actually, since the beginning of red clover being used in South Africa, it has only been the annual type that has been available. But these have a poor system fit with perennial multi-species pastures. Some years ago, actually sort of 3 years ago, through a visit from a professor from Mendel University in the Czech Republic, we became aware of grazing types of red clover. And we have started evaluation of these grazing types. And obviously we evaluate yield, but also persistence and other characteristics for improved system fit. So the first thing that we look at, the characteristics that are relevant in multi-species pastures in terms of complementarity, is they must be deep-rooted. They must be taller growing than white clover, which is why we like the red clover. The red clover also does not become dominant, but it is more competitive than Lucerne, which is the other alternative legume, and it combines well with the other components. It obviously fixes nitrogen, but importantly, it shares more nitrogen with its other components than Lucerne does due to its root system structure. And there's more milk production from red clover than there is from Lucerne because of the enzyme polyphenol oxidase, which causes improved or increased bypass protein. So the types and cultivars we evaluated or are still evaluating. So obviously the annual type is our control. Whoops, sorry. And then we have the grazing types which have been developed in New Zealand and the USA, which are this group of cultivars. And then there are the grazing types from Europe, which actually originate from collections out of meadows that have been used in Switzerland for more than a century. So if we look at some of the data, and here I just want to focus on the different groups of cultivars. So the green, the ones marked with green, are the group that is the New Zealand US group, and the other ones are the European ones. So for the first winter, it was basically the New Zealand USA grouping that had the highest production, and the European ones were basically winter dormant. If we then looked at early spring, in the first year, we saw that some of the European ones actually started moving up into the top group, which was an important result. But some of the European varieties were still not matching up in terms of yield. But if we look at the second year, early spring, then all of a sudden we see that it's basically the New Zealand USA types, and the European types are nowhere to be seen. So the problem with that is that the breeders did say the European types like a longer grazing rotation, which may not fit the grazing rotation that we use. So we use a shorter rotation than they use, which these cultivars then did not tolerate. In the summer, it was a good mix. They were doing well. Both types were doing well, or some of both types. And then a really important consideration is ground cover. Because in the multi-species pastures, if we have bare ground, we get weed ingression. So ground cover is a really important measure. We looked at percentage green leaf 10 days post-harvest. And these are, for instance, some of the results that we got. So what was important is that the European, or some of the European types, have a really good ground cover from like immediately post-grazing. So that would mean we can limit weed ingression. And this is what it looks like. So for instance, this is one of the European types, and this is the USA type. So you can see there's a marked difference. And we actually found the same result under grazing and under cutting. So in summary, winter production, up to now, only the New Zealand USA cultivars showed significant yield. Early spring was mainly the New Zealand USA types with some European types, but only in the first spring. In the summer, the EU types were better. The tallest cultivars in spring were the New Zealand USA types, which is important because the other pasture components during that spring growth period are also quite tall. And in summer, the EU cultivars actually also reached the required height to complement the other components. And the superior ground cover was really only achieved by the EU cultivars to try and prevent weed ingression. So in conclusion, to extend the production period of red clover, we think it is best to mix a New Zealand USA type with an EU type cultivar and basically use a blend of cultivars to improve the production period. To improve the ground cover, we have to include EU cultivars to help us manage the weed ingression. And for temporal complementarity, we should avoid mixing the annual type with the grazing type, because otherwise we will again end up with open spaces. Thank you very much.
Thank you very much, Sigrun. Before you run off, there is time for one question also for you. So is there a question in the audience before Sigrun actually escapes the questions. There's one question, Richard. Oh, it's coming.
Hi, Sigrun, thanks for a nice talk. So the grazing type is a perennial then?
Yes, it is perennial. Yes.
Okay.
Or as perennial as, we don't know yet its extent of its perenniality because we haven't really reached the end of the trials yet.
Yeah. Of course, I saw some data on like perennial, the difference in root production between perennials and annuals from like say perennial wheat versus annual wheat. It's a massive difference in root production. And I wonder if it's the same with these, because in terms of carbon storage, that sort of thing for your soils and improving soil health, it might be good that the perennials have a better effect on your soil.
Yes, we're hoping they will. Towards the end of the trial, we will hopefully be able to dig up some plants, which we now can't do at this point, but we might also do that once we have them in the grazing trial. So it's a good point. So I do think the root, that's very important for us, is that there is sufficient rooting in the profile as well. Yeah. And it helps with water use efficiency as well.
Thank you very much, Sigrun. Thank you for your presentation on enlightening us how we can use them in the multi-species of the different red clovers and how they can be beneficial. So that brings to the end of the 15 minutes presentations. We are now starting with the standard posters, which will be 5 minutes per presenter. So the first presenter for us who will be enlightening us about morphological and physiological responses of Lucerne. It's Medicago, Sativa is genotypes in soil acidity, will be Sesethu Sokoko. Yeah. Okay. There's this one.
Good afternoon, ladies and gentlemen. I am Sesethu Sokoko. This is one of the work that we did over the previous year where we wanted to check if how Lucerne genotypes respond to soil acidity. We were mainly focusing on the morphological and physiological responses of Lucerne genotypes. The reason we wanted to do this is because Lucerne is one of the mostly cultivated leguminous crops in South Africa and all around the world. However, it cannot tolerate soil acidity, but it has a wide variety of adaptation to other agroecological conditions. Now, we were able to attain this information by using this methodology where we planted 6 different Lucerne genotypes in 3 pH treatments. We had pH 4, 5, and 6 as the hygromix. So in this trial, we wanted to check if how the entire shoot development respond to the stress. And therefore we looked at the stems and the branches and the shoot height. And this is that entire shoot growth. We then at the later stage, when the plants were reaching maturity, we checked the physiological traits where we wanted to see if the plants are exposed to the stress, how is the cell membrane responding? And then we did that using the electrolyte leakage test. And we also looked at the hydration and dehydration of the plants once it is exposed to the soil acidity. And at the later stage, we also detect the turgidity mass. The other thing that we looked at was the entire root development after we had planted them for all over for a year in the plastic bags. And to my surprise, some of the plants had nodules, which is very rare when you plant a legume in acidic soils. We are then able to look to find the information on the morphological characterization. Some of the lines that had good performance had a better shoot development than the lines that were not able to withstand the stress. And one of the lines that performed great is the one that we are currently breeding at the ARC. We then looked at the physiological traits where we wanted to check the cell injury. So the ones that had an enhanced electrolyte conductivity showed that there is a leakage of ions. And this means that if we expose the plants to the stress, there is a lot of damage that occurs to the cell membrane. And that also led to leaf dehydration, because when the plant is exposed to an abiotic stress that it cannot withstand, then there is a permeability on the cell. And at the later stage, when we look at the root development, we focused on the root length, because Lucerne is a deep-rooted plant. We wanted to see if how long can it grow when exposed to the stress. And then the ones that had, the ones that were able to withstand the stress had long, extensive roots, but the ones that were hindered under the stress, then they had shortened roots. However, in this study, we want to further screen these lines, because there hasn't been much variation when we did the statistical analysis. So we want to take the lines further into the field experiment, where we will then be able to check if in a prolonged time, what's going to happen to the genotypes. But our main focus are the two lines that are being screened at the ARC. For if ever we are able then to select the ones that can withstand the stress, then we'll be able then to incorporate them in our breeding programs. Thank you so much.
Thank you. Thank you, Sesethu. I have time for one question for Sesethu, a very quick one. A hand at the back.
Thank you for a nice talk, Sesethu. I just want to know, as a non-pasture scientist, why doesn't Medicago sativa tolerate acidity?
That's a good question, but most leguminous crops are not able to grow in soils that have pH that is less than 5.5. So currently we are trying to screen and have one cultivar that is going to grow in those type of environments without having to incorporate the soil with soil supplements. For instance, these lines were evaluated here in Sidara, because Sidara experiences a lot of rainfall. So it's something that we are trying to see if it's going to be possible going forward.
Thank you so much, Sesethu. Yo, you were quick to run, eh? And then our next presenter for today is Mihle Sokupa, who will be presenting about Rhizobium, and then she will finish the rest.
Good afternoon, everyone. I think we might all be familiar with the proverb that says, take care of the pastures and the animals will take care of themselves. So my name is Mihle Sokupa, and I'll be presenting today part of my PhD work, which is titled Rhizobium leguminosarum biovar viciae enhances growth and yield parameters of veg varieties. Legumes like veg play a vital role in as supplement feed during the dry season, where rangelands cannot sustain livestock dietary needs. Veg is a high-yielding legume that adapts to a variety of soil types. However, these high-yielding legumes are sometimes affected because their growth is affected because, though these are high-yielding legumes, their productivity is hindered because they are limited by the fact that there are no compatible Rhizobium strains in the soil, or sometimes temperatures also hinder these legumes. Rhizobium leguminosarum biovar viciae are bacteria that have been made specifically for peas and veg. They increase nodulation, which improves the mechanism of biological nitrogen fixation, which takes us to why we did the study. The objective of the study is to determine the effect of different inoculation levels of Rhizobium leguminosarum biovar viciae on growth, biomass yield parameters of veg species. The study was conducted at the Northwest University in Mafikeng during the two winter seasons of 2023 and 2024. Soil samples were collected from surrounding areas of Mafikeng. They were mixed and analyzed for pH and minerals. The experiment was carried out in a pot medium in an open field. The seeds were randomly planted in plastic pots filled to identical soil weight. The treatment factors were 4 veg varieties, Morava, common veg, Namoi, and Haymaker Plus, and 5 inoculation levels, which is 0, 150, 250, 300, and 350 mils per 50 kg bag of seeds, with 20 treatments in total. The treatments were replicated 20 times, resulting in 400 pots. Sowing depth was 4 centimeters with a sowing rate of 25 kg per hectare. The pots were irrigated weekly. 12 plants were randomly selected from each treatment, where plant height, number of leaves, number of branches, chlorophyll concentration was measured fortnightly. Above ground biomass yield was harvested at 60 days after planting. The data was subjected to a 2-way ANOVA for SAS 2010. It's quite a lot of data, but I'll try to be as brief as possible. In 2023, Namoi and Haymaker Plus had the highest number of leaves, while in 2024, Namoi only had the highest number of leaves. And then when we compared each variety in both years, we saw that level 250 and 300 produced the highest number of leaves in all four varieties in all weeks. Then this table shows the plant height. Also here, we saw that in 2023, Namoi and Haymaker Plus both had the tallest plants, but in 2023, we saw that only Haymaker Plus had the highest plants. And then when we compared each of the varieties in both years, we saw that level 250 and 300 produced the tallest plants. This is the results for all the weeks from week 2, week 4, week 6, and week 8. And then this table shows the number of branches. Number of branches in 2023 and 2024, Namoi and Haymaker Plus both had the highest number of branches. And in 2024, both of them, they still had the highest number of branches. And when we compared each of the varieties in both years, we also saw that still 250 and 300 mils per 50 kg of bag of seeds of Rhizobium inoculant was higher. And then we also noted that beyond 300, there was no effect, or it did the effect of Rhizobium did not increase the number of branches. And then for chlorophyll concentration, we did not see a specific trend when we look at the chlorophyll concentration in all the weeks, but we did see that when comparing the uninoculated and inoculated plants, we did see an improvement, even though there was no specific trend as to say it increases up until when, because in some weeks we saw that the chlorophyll was higher, and then in the next week it was lower, then in the other week it was higher. So there was no specific trend in terms of chlorophyll concentration. Then this table shows the effect, the biomass yield parameters, where we measured, where we harvested for fresh weight, dry weight, fresh yield, dry matter yield, and crude protein yield per hectare. Here we saw that fresh yield and fresh weight, Namoi had the highest fresh yields and fresh weights in 2023, while Haymaker Plus and Namoi both had the highest yield in 2024. And when we compared them in both years, each variety in both years, there was no significant difference, meaning the effect of yield did not really matter. And then for dry weight and dry matter yield, we saw that Namoi and Haymaker Plus both had the highest dry matter yield and dry weight, and produced the highest dry weight and dry matter yield at level 250 and 300. Beyond that, which was 350 mils per 50 kg bag of seeds, there was no effect. And then for the crude protein yield, we saw that Namoi and Haymaker Plus both had the highest crude protein yield per hectare in all of the varieties. In in the current study, the number of leaves, plant height, and number of branches were improved by Rhizobium inoculation in both years. The highest percentage increases between uninoculated and inoculated plants was received in 2023, where we had 23%, 30%, and 22% for number of leaves, plant height, and number of branches, respectively. An increase in the morphological parameters ensures high biomass yield in plants, which is good for livestock production. The improvement in the inoculated plants could be due to the effect of Rhizobium bacteria on biological nitrogen fixation. The highest percentage increase in chlorophyll concentration was 23% and received from Namoy in 2023. Various authors also reported an increase in chlorophyll concentration in some of the legumes that were inoculated with specific Rhizobium strains. However, did not have a uniform trend between varieties, inoculation levels, or the days of data collection. The differences in the chlorophyll concentration could be due to the temperature of the days. It could be due to the temperatures on each specific day of data collection. Fresh weight, dry weight, fresh yield, dry matter yield, and crude protein yield per hectare were improved by Rhizobium inoculation in the study. These results concur with Alberg et al. 2004, who reported an improvement on biomass yield and crude protein yield of common veg planted in a field. However, they reported higher results as the highest dry matter yield that they reported was 6,000 kg per hectare, while in this study we got the highest dry matter yield as 1,900 kg per hectare. The Rhizobium strains improve nodulation, which increases photosynthetic ability of the host plant. Namoy and Haymaker Plus perform similarly in the study because they both fall under the viciae villosa subspecies, while common veg and morava also perform similarly, but though they were lower because they fall under viciae sativa subspecies. So these among the four subspecies, among the four varieties that we use, 2 fall under one subspecies and then the other 2 fall into another subspecies. So we did see that indeed the 2 are similar and the other 2 are similar. In conclusion, the results show that the parameters measured in the study are indeed improved by Rhizobium inoculation. Namoy yielded the highest results in most of the parameters, though in other parameters, it was either Namoy and Haymaker Plus, but Namoy had the highest. And then Rhizobium inoculation beyond 350 mils per kg bag of seeds decreased or was ineffective in the yield of these veg varieties grown in semi-arid regions. Thank you.
Thank you. Thank you very much, Mihle. Unfortunately, we don't have time for questions for you. Anyone who has a question for Mihle can engage with Mihle afterwards. Our next speaker for today will be Fortune Manganyi-Valoyi, who will be presenting on the evaluation of a smart grass Bracharia species.
Okay, thank you, Kuli. Good day, everyone. My name is Fortune Valoyi, and I'll be presenting some of the work that we are currently doing at the ARC, where we wanted to evaluate the climate smart Bracharia accessions. So we all know and preach feed shortage and how it has a negative effect on the livestock production. However, climate smart forages can save as an alternative to fill up the gap. And Bracharia grass species are classified as climate smart because they are able to adapt in drought resistant and also in infertile soil. So we wanted to evaluate the adaptability and the agronomic characteristics of Bracharia grass accessions for their potential as a forage resource. So the experiment was carried at the ARC Rodiplat, whereby a total of 27 grass accession replicated 4 times were evaluated for their potential under irrigation and dryland condition. And this accession consisted of 3 cultivars of Bracharia brisanta, 3 cultivars of hybrid, 3 cultivars of humidicola, and 4 cultivars of Microbetata species. Seeds were germinated in seedling trays and then transplanted into 2 by 4 meter square plots with 12 plants per plot, where data was collected at 75% flowering and sample harvested for biomass determination. So with the result that we got for Bracharia brisanta species, from year 1, they produce at an optimal level, and then they increase in year 2, and then again decline in year 3 for both irrigated and dryland condition. Species such as Zarex 730, 473, they produced more dry matter for the consecutive of 3 years. And when we move to Bracharia hybrid, which is more similar to brisanta, they also produce at optimum level in year 1, increases at year 2, and then decline again at year 3 for both irrigated and dry land condition. However, these are still within the optimum production that is needed. And then we move to Bracharia humidicola. These are creeping species, very suitable for grazing. They also produce high in the first year, and then they decline as the year goes by, with Larnero producing high compared to the other species. The last cultivar is Bracharia micropedata, which also produces high during the first year, and then they decline as the years goes by, with Kufa being the highest produced within the species. And with this result, we have noticed a significant increase in dry matter yield in year 2 in most grass accessions, such as cultivars under brisanta and hybrid. These can be attributed to the nutrient enrichment of root crown in the first year, then aggressive growth in the second year. And for this reason, we concluded that Bracharia grass accession have the potential to serve as an alternative resource because they can grow in both irrigated and dry land condition. However, most studies, such as looking at their nutritional value and how that affected the animal performance, needs to be evaluated. Thank you very much.
Thank you very much, Mevaloyi. One question for Mevaloyi. A hand there, is Onati.
Okay, thank you for your presentation. I heard you mention a fluctuation in terms of dry matter production, a difference between year 1 and year 2 where there's a decline. What could be the reason of the fluctuation in terms of the dry matter production of these Bracharias?
Okay, thank you for that question. The aim of the study was to check whether they can be able to produce in the different years. So with Bracharia brisanta and hybrid, they produce more on the second year because on the first year they invest mostly on the ground, on the root crown. And then species where such as Bracharia humidicola and Microbetata, because these are grazing species, they produce more on the first year. So we wanted to check how is the production throughout the years so that we can be able to recommend some cultivars for farmers.
Thank you very much, Fortune. We can give Fortune a round of applause now. She was eager for another question, but we can save that later for discussions. And then our next speaker is Noluvo Joyi, who will be enlightening us on the influence of nutrient-rich pig slurry and legume inclusion on forage productivity. This one or that one?
Greetings, everyone. Today, we are going to learn about influence of nutrient-rich pig slurry and legume inclusion on growth performance, productivity, and chlorophyll content of Bracharia brisanta varieties by Noluvo Joyi and Mthunzi Mdela. Climate variability and rangeland degradation threaten rangeland production, leading to a poor animal performance and productivity. Hence, there is a need for alternative forage reserves to complement rangelands. However, to maximize forage productivity, improved forages demand fertilizer application, which is costly for small-scale farmers. Animal manure, e.g. pig's lard and legume, can improve soil fertility and enhance pastures productivity. Pig's lard is rich in phosphorus, organic matter, and other essential minerals. Thus, we asked, what is the optimal rate of pig's lard that can maximize the performance and productivity of Bracharia brisantha? Secondly, does intercropping with a legume enhance the performance and productivity of Bracharia brisantha? The study was conducted at Manganyi-Valoyi. The study was conducted at Manganyi-Valoyi at University of Fort, where a 2x2 by 4x4 factorial pet experiment within a RCPT was used. The two varieties. Two Bracharia varieties, Marando and MG4, were planted with or without Labdaparas. These pasture type were assigned. These pasture type were assigned to 0, 25, 50, and 100% pig's lard rates for 4 consecutive applications at 30 meters cubed per hectare. The total concentration of pig's lard was 100%, downscaled to 75:25 and 50:50%. Tiller production, chlorophyll content, and biomass production were determined. The four-way interaction was determined using a general linear mixed model at P less or equal to 0.05. The chlorophyll content was measured using a spot meter and a tiller production by counting. Unfortunately, we couldn't present the results for legumes because there was no significant interaction. Table 1 shows a significant interaction between pig's lard application time, monoculture, and varieties. At 0%, there was no significant interaction between these. Yeah. However, at 25%, we observed a significant increase for tiller production in monocultures after 4th application, with MG4 having higher tiller production. At 100% rate after 4th application, MG4 had a higher tiller production, even for mixed. Okay. The four-way interaction showed a significant increase in tiller production, where 0% had no significant effect between mixtures and monocultures for both varieties. At 25%, we observed an increase in monocultures for MG4. Even at 100%, MG4 obtained higher tiller production, even for mixtures. And if we can see that for MG4, we had 30+1 tillers for plant. Even at mixtures, we had 43, which means that it is indeed to get higher production, grass species need to be intercropped with legumes, because we can see the difference between monocultures and mixtures.
Monocultures had 30 tillers, while mixtures had 43 tillers. Hence, if farmers want to have a higher production, they should opt up for MG4 because for a higher plant density and a Figure 1 showing biomass production of Bracharia presenta across varying peak slaughter rate. There was a significant difference between PSR and varieties. The biomass production increased linearly from 0 to 100%, with MG4 having higher biomass production compared to Marandu. However, Marandu obtained a similar biomass production at 50% and 100% rate. Figure 2 showing chlorophyll content of Bracharia presenta across varying peak slaughter rates. After first application, there was no significant application of peak slaughter. There was no significant application between chlorophyll content for both varieties. However, after second application, there was an increase in chlorophyll content with 25 and 50% having higher chlorophyll content compared to 0 and 100%. There was a decline after third application in chlorophyll content. Then after these results show that as the plant grows, the crude fiber increases. After fourth application, there was a slight increase in chlorophyll content. This could be because repeated application of peak slaughter result to regrowth and new cell tissues. In conclusion, the results of the study show that peak slaughter improves productivity and performance of Bracharia presenta. Therefore, pasture establishment can be conducted via application of peak slaughter to increase forage productivity to complement rangelands. Hence, it is recommended that farmers apply peak slaughter at 100% rate. I'd like to thank you, GSSA, for sponsoring me, University of Fort Hare. Thank you.
Thank you very much, Noluvo, for enlightening us on how peak slaughter can be beneficial to Bracharia. It takes a lot of effort to stand in front and still continue with your talk, so I applaud you. You did very well. And then our next presenter for this session will be Uleseho Majecha, who will be presenting on altering forage legume planting dates to improve biomass productivity. Is Uleseho in the audience? Okay, so unfortunately, Lesejo is not here today. Please, I do not deserve an over for that. Lesejo is not here today, so therefore we can move on to the next presenter, who then is Donna. Is Donna Bertak in the audience also? Okay, so Donna will be presenting on comparing 2 years of forage legumes and winter cereal mixtures on Sedara Research Farm Station. You need this mic or that mic? That's laser next, and then if you want to go back.
Okay, afternoon everybody. Okay, can everyone hear me? All right.
More closer to the mic, please.
This one or that one?
Anyone.
Which one's better? That one. Okay. Okay. All right. This is just a snapshot of some trials we've been working on. So 2 years of forage legumes with cereal mixtures at Sedara. So basically the idea behind it is that if you put a legume in a mixture, then it'll improve your protein content And then the grass gives you the energy component. And then it could also increase the dry matter content compared to the grass alone. So in 2023 and 2024, 2 cereal legume trials were planted dry land in March. The first trial was a grazing vetch with winter cereals. So the cereals were black and white oats and stewling rye. And this was cut to simulate a grazing trial, so multiple cuts. And then the second trial was forage peas and winter cereals. And that was ran to simulate a silage trial. So you wait until the peas are starting to pod, and then and try and match it to the grass. Okay. All right. Unfortunately, we couldn't get the exact same varieties each year, but we tried our best. Okay, so this is the vetch and cereal trial. 2023 is on the left and 2024 is on the right. So 2023 had better yields than 2024. There were 3 cuts in both instances across the season. Where in 2023, the mean yield was about 5.4 tons of dry matter per hectare, where in 2024, it was only about 3.75. And then the highest yielding in each trial, 2023 was about 6.57, and in 2024, only 4.76. What was interesting, is that the 3 top yielders are different mixtures. So it's a vetch with a white oat, it's a vetch with a stewling rye. I think that's a different stewling rye. And in general, the mixtures do slightly better than the grasses on their own. Yeah, in 2024, it's also spread out a bit, the top 3, but it's a little bit less varied. Okay, so then the forage peas and winter cereals. So the 2023 and 2024 overall yields were a bit more similar. There were only 2 cuts per season. So the first cut is the silage cut, as I said before. And then the second cut the peas don't survive, so it'll only be grass. So then in 2024, it was a bit more even, but a lower yield in the second cut. And then 2023, it was interesting, there was a bit of a grouping here where the white oats sort of added a lot to that mixture. So the 3 best were all white oats mixed with different peas. Okay, so the mean total yields were both around 4 tons per hectare. And then the highest total yield was a little bit higher for 2023 than 2024. Okay, I was trying to keep this short, so I didn't add in the rainfall data, but basically what we think happened was that the higher yields, especially for the vetch and the cereal in 2023 over 2024, was possibly due to, we had really good rains in May that year, and so that helped the sort of winter cut, the second cut, which was a lot better in 2023 than 2024. And then in the pea and cereal trial, in some instances, the peas really dominated the yield. So then in the second cut, the grass actually was a little bit swamped and it didn't come back as nicely. But in conclusion, the mixtures of legumes with winter cereals is a viable option for dryland production, especially in the KZN Midlands, if as long as you plant before the autumn rains stop. Okay, thanks.
Thank you very much, Donna. Unfortunately, we don't have time for questions for Donna. You can engage with Donna later during the networking. And before the next speaker comes in online, apologies, Lesego is available. She is online, so she'll be presenting from online. But before she presents, I just have an announcement to make for the ladies. There is someone who forgot her phone in the bathroom. So ladies, can you please check if your phone is still with you? And if not, then you can come check if this is your phone or not. So then our next presenter will be an online presenter, which is Lesego Mashika, who will be presenting on alternating forage legume dates to increase biomass productivity. I'm not sure if they can be able to join her online. Oh, and ladies, when you do come, you might have to just prove that it is your phone by unlocking the phone so that we know that it's going back to the rightful owner. Okay, thank you, Lesego. You can go ahead. We cannot hear you, so I think you are muted on your side. So can you try maybe unmuting so we can maybe hear you? Or just
okay, it seems like I'm an unmute. I'm now okay.
Yes, you are now okay. You can go ahead with your presentation.
All right, let me put up the presentation. Can you see the presentation?
Yes, we are able to see it.
All right.
Can you please just make it on slideshow?
Okay, I think I'm good now. And greetings, I'm live from University of Limpopo. My name is Lesego Mpanza. Going to share with you part of my PhD study on altering forage legume planting date to improve biomass productivity and resilience. Lack of good quality and quantity forage limits ruminant productivity and induces rangeland degradation in livestock production system as depicted by Figure 1. The ongoing climatic changes, such as temperature shift and precipitation variation, have a significant impact on forage production. And as depicted by Figure 2 and Figure 3, cool season annual clover and veg species are the legumes well known for production of good quality forage, particularly the protein. Planting date and species diversification are some of the agronomic practices for manipulating the seasonal distribution of forage availability. The aim of this study was to assess the effect of planting date on biomass production of annual clover and veg cultivars grown in no-till system for climate change adaptation for fodder production. And a field experiment was laid in a split plot design with 4 replication, and it was conducted at the University of Limpopo for 2 growing seasons, the 2018 and the 2019. The treatments were as follows for this split plot. We had 3 treatments, 2 planting dates, which is the June and July planting date. Then we have cultivars of annual veg and annual clover. Each species had 2 cultivars, and they are outlined on the screen. I believe you can see them. Then we had the 4 harvest stages. These harvest stages were mid and late vegetative stage, as well as flowering initiation, But for this study, I'm going to present biomass production at 50% flowering, which was harvested from a 1 meter by 1 meter quadrat, dried to constant weight, and weighed for determination of the dry matter accumulation. And the data was subjected to ANOVA using the SARS 9.4, and the post-hoc analysis was done using the LSD at 5% level of significance. Coming to the results. The results are indicated in table 1, which shows the effects of planting date and cultivar on biomass production of annual clover and veg cultivars harvested at 50% flowering. According to these results, the table indicates that the planting date, the cultivars, as well as their interaction, had a significant influence on production. With regards to the planting date, the July planting date consistently produced significantly lower biomass production in the 2 growing seasons when you compare to the June planting date. And the cultivars had differed significantly in terms of their biomass production. And biomass production ranged from 996 kilograms per hectare to 3,821 kilograms per hectare in the 2018 season, while in 2019, biomass ranged from 958 kilograms per hectare to 3,713 kilograms per hectare, with Rhizobium performing significantly being the highest producer, and Rhizobium is a Persian clover cultivar. Opolska being the significantly lowest producer, and Opolska being our crimson clover cultivars. These cultivars were selected, some are from Germany, Europe, and some were selected from South African seed market. And we, according to the seed producers or the seed market sellers, they indicated that the South Africans ones are the ones that have been long available in the country. Then the results indicate that in cultivars are good. Cultivars, Rhizobium as the best and Opolska as the lowest. Regarding the conclusion, the June planting date consistently produced higher biomass as compared to the July planting date, possibly due to the longer growing seasons. And Rhizobium, Alex, Eliza, and Elite, added with Dr. Bowman's, are the highest producers. And know that Rhizobium as well as Laser are our Persian clover. Alex and Elite are our Bersim clover cultivars, with Dr. Bowman's being a hairy veg cultivar. The lowest producers were Capello, Hanka, Timok, Linkaras, and Opolska, which are a mixture of crimson clover as well as Capello as the hairy veg. It is therefore critical to choose the optimal planting date and cool season pasture cultivars which have high thermotolerance adaptive capacity to maximize forage production and reduce feed gaps. Thank you very much.
Thank you very much, Lesego, for presenting to us on how alternating planting dates could be beneficial for biomass production. Since Lesego is online, I'll give time for one question if there is really a burning question in the audience. Is there a question from the audience? Okay, there's no question from the audience. So thank you very much, Lesego, for your presentation. Can you please give her the last round of applause? And then the next presenter for today is Rebecca Ellerbeck, who will be presenting on the effects of utilizing a cover crop under different grazing pressures. all yours. Do you prefer this one or that one?
There it is.
Thank you so much. All right. So hello, everyone. I'm Rebecca. I'm from Stellenbosch University, and we're busy looking at different management strategies for utilizing a cover crop. But why graze a cover crop, and how does it differ from planted pastures or rangelands? Well, cover crops are very similar to planted pastures, except that they're not actually intended for grazing. They're planted not by livestock farmers, but by crop producers who cultivate them in order to benefit the soil. They're usually only planted for a single season within a cash crop cycle or a rotation. And unlike a cash crop, they aren't harvested, so they generally don't generate their own form of income. So this is a challenge in planting cover crops. And by integrating livestock to graze the cover crop, we can generate a cash flow for the system. But because the purpose of the cover crop is primarily to benefit the soil, good grazing management is necessary to make sure that the cover crop isn't damaged to the point of losing its functionality. But what is good grazing management? So we're looking at 4 different systems, each comprised of a single hectare, each planted with the same number of animals, stocked with the same number of animals, and planted with the same cover crop mixture, but either left as an open paddock or subdivided into multiple paddocks. So we're comparing continuous versus increasing levels of rotational stocking. So when we add our animals, we're using sheep. We have the same stocking rate, but an increasing stocking density across treatments. And the grazing period is managed such that the 8 paddock system moves twice as fast as the 4 paddock system, and so on. This way, all of our sheep complete a grazing cycle simultaneously and can start again together. We also have a control that remains ungrazed throughout the trial. Our measurements focus on economics, on soil parameters, on herbage production, and on livestock performance. When we look at the livestock for the first season, we saw no significant difference in sheep weight across treatments, but we did see that treatment 4 with 8 paddocks had a significantly slower growth rate compared to treatment 2 with 2 paddocks. When we look at forage availability, again, we saw no significant difference in average availability between treatments, but all of our grazed treatments showed significantly more forage availability compared to the ungrazed control. Another note on forage availability is that it was generally very high throughout the season. We often saw a higher forage availability after the sheep had finished grazing a paddock compared to before they started, meaning that the forage was growing faster than what the sheep could consume it. This led us to theorize that the lack of significant differences observed could simply be due to understocking. And we concluded that our cover crop is definitely not being utilized to its full potential under 8 sheep per hectare. Now, granted, this data is only based on 1 season's worth of results. We're still busy looking at soil, at nutrient composition, at species composition. But yeah, this brings us to the final question, what's next? And we're essentially repeating the same procedure as last year, just with more sheep. We're increasing now from 8 sheep to 12 sheep a hectare for this season. And we're keeping the stocking period the same to retain continuity between last year and this year. So there's a lot more detail on this. I really wish I could talk your ears off, but please come chat to me afterwards. And thank you for your time. Thank you for listening.
Thank you very much, Rebecca. And indeed, we'll see you next year. Luckily, it's in Cape Town, so you're very close by. You will do a full-on presentation that time. Is there one question? Okay, I'll allow the 2. Us and then us first, and then you, Richard.
Yeah, lovely presentation. Question though, are you not confining season effects with stocking rate effects by increasing from 8 to 12 next year? What's the reason for doing that?
Yes, it's a very good question. So from the beginning, we planned to anyway analyze them as 2 different experiments because of climatic data. It would anyway be challenging to analyze the data from last year and this year as one single continuous experiment. And ideally, I would love to keep the stocking rate and everything continuous between the seasons. But based on what we learned last year, it didn't make sense that we've seen a problem to continue with the same problem, where we can take what we learned last year and realize, okay, we realized that our stocking rate was too low. This can support a much higher stocking rate. So the logical next step is increase the stocking rate and see what happens. So it's definitely not an exhaustive study. There's so much more that we actually want to look at, and so many other answers we want to get. But in terms of just the logical next step of establishing how does grazing on cover crops work, we thought it's going to be more valuable to increase the stocking rate for the second season.
Thanks, Rebecca. Very interesting. And it's actually what a lot of studies have shown, that when you increase the number of paddocks, the weight gains of your animals go down. Because what's happening is you as I was presenting this morning, you're allowing the grass to grow out in the multi-paddock systems. So you're putting your animals into taller, lower quality grass. Whereas by keeping them for a longer time in the two paddock system, they're more able to keep shorter and eat the regrowth of that material. So I think, yeah, increase the stocking rate, that would be good as well. But definitely, I think from a theoretical point of view, I see that the two paddock system should give you the better animal weight gains. Yeah.
Thank you so much.
Thank you very much, Rebecca, for your presentation, and hopefully we'll see you next year. And then Kwena Mokoboki is unfortunately not here to present, so then we'll be skipping to the next presenter, who will be our last presenter for the day, because also Peggy Karimi also is not available to do a presentation. So coming to bless us again for the session is Dr. Thamsanqa Mpanza for his presentation on the fermentative profile and aerobic stability of Bracharia.
Thank you, Program Director. To preserve time, let me just go here. We know that food shortage is a critical issue during dry season period. So we look at a means of preserving it in a form of silage. However, a fermentative characteristics of a monoculture and salt material is affected, or is not fermenting well in terms of pH. So mixing it with other ingredient, the research shows that it optimizes the fermentation and enhance aerobic stability. Therefore, in this study, what we collected Bracharia, same one that was presented by this one, but without separating it into species. We just collect as Bracharia, then we mix it with Mihle Sokupa, which is an invasive plant that has high protein, just to see how it can enhance the fermentation. So I'm not going to dwell much in this because the study is the same way the Bracharia was produced. But what I want to indicate that the inclusion of Mihle Sokupa, it was 20% and 30%, 0% being the control, so that we can be able to see how it's going to affect the production. Here it was, that is where we were milling it. This is how we ensile it. The most interesting thing is that the pH drops from, this is the day of ensiling. Normally the pH is above 5, but in 3 days it were able to drop into below 5, which means that it fermented. But what we noticed that in day 40, after ensiling, it drops, but it's only the one that were treated with Mihle Sokupa that dropped the pH to 4.27 and 4.3, which is close to this range of a well-fermented forage. But when we look at the aerobic stability, which is the exposure of silage when after ensiling, when you open it to feed it to animal, how long does it take for the silage to maintain that quality? So also we measure the pH. You can see that as the days increase, this is when we open at day 40 here. Then this is day 2, day 4, up to day 8. But you can see that up to day 6, you can still feed that forage because the pH is not yet above 5, which we use as a cutting off pH value. Then at day 8, the pH started to increase to above 5. However, we're interested to look at what is the cause of this statistical change here to these that were supplemented with Melia azadirachta. So things such as looking at the volatile fatty acid, ammonia, nitrogen, even look at the microbial composition as well as microbial composition, so that we can be able to look at the microbe to see that is there any microbes to follow that are of interest or what. So basically, in this study, we concluded that Mihle Sokupa included to Bracharia up to 20% to maximize or optimize the fermentation. However, we needed to do a lot of study to look at the microbial composition, because normally we tend to associate forage silage quality with the fermentative characteristics, which is the pH dropping, which is also leave the microbial composition, which plays a role to the quality of the silage. Thank you.
Thank you very much, Dr. Mpanza. Is there a question for Dr. Mpanza in the audience? Oh, there is one hand at the back.
Thank you very much for the nice presentation. Is Melia azadirachta not poisonous in some or other way to the animals?
It depends. If you look at the leaves, because sometimes we confuse the toxic substance is more on the seed itself than on the leaves. The leaves are safer to feed the animals. Of course, because it's a plant, it has plant secondary metabolite, which governs how much you can include it to animals without having a negative effect to animals.
Thank you, Dr. Mpanza. Can we please give him a last round of applause? And then I would like to thank everyone for sitting through this day and through this session, which was the last session for the day. The last presenter, as I have mentioned, which is Ubegi, will not be here to present, but I was made aware that they have a poster. So if you'd like to see what is happening in the upper and lower Midlands of Kenya, you are more than welcome to check the poster out to read what is the content of it. So thank you, everyone. This is the end of our presentations for today. But before you go, can you please give all presenters a round of applause?